days, and Ada has been working with that stuff. So I think that she is one of the pioneers in this field, and maybe she's going to tell us about those secrets that life has for us, discovering how the structures can tell us about the gene code evolution. Thanks, Ada, to be here again. Bien. Todo mundo tiene en sus manos, espero, una semblanza de la doctora Jonat. Yo nada más quisiera remarcar algunos hechos que son muy importantes en su carrera. La doctora Jonat es realmente un ejemplo del esfuerzo. Ustedes podrán leer en la semblanza pues que para ella la vida no fue nada fácil, como no lo ha sido para bastante de nosotros tal vez, pero ella con su esfuerzo, con su dedicación logró ser un premio Nobel. Como ustedes pueden ver en la semblanza, ella estudió química en la Universidad de Tel Aviv y después hizo una maestría en bioquímica para posteriormente hacer un doctorado en cristalografía de rayos X en el Instituto Weizmann, donde actualmente trabaja, y ella sigue haciendo investigación. Me estaba platicando hace un rato que ha dejado de hacer cristalografía porque es más complicado, pero está usando la microscopía electrónica para revelar estructuras, aunque con menor resolución. Ha sido muy útil para empezar a descubrir nuevas cosas que tienen que ver con nuevos blancos en el ribosoma para antibióticos. Como ustedes lo verán ahí, también es muy claro cómo este ingenio de la doctora Jonat fue importante para lograr cristalizar los primeros ribosomas. Había un problema en ese tiempo y era que los ribosomas, como son complejos supramoleculares, tomaban mucho tiempo para estar irradiando y esto llevaba a su desintegración. Finalmente, ella y otros colaboradores desarrollaron un método de la cryo-cristallografía de rayos X, donde pudo evitar la desintegración y estar bombardeando por más tiempo para llegar a resolver la estructura tridimensional del primer ribosoma, que publicó ya en el 2000. ¿Qué más les puedo decir acerca de un premio Nobel? Ella tiene todos los honores y ha sido profesor emérito de muchas universidades en el mundo, y nosotros la hemos tenido aquí, esta es su tercera ocasión aquí en la facultad, y como parte de todas estas visitas y como parte del esfuerzo que se hace en la facultad por traerles el conocimiento de primera mano a los muchachos de alguien que está haciendo ciencia, que no ha dejado de moverse en esa dirección, pues la traemos aquí, y además ha pasado a ser una profesora extraordinaria de la UNAM adscrita a la Facultad de Química. La Facultad de Química paga su salario, por eso está aquí la doctora, pero no solo por eso, a ella le entusiasma muchísimo hablar con los estudiantes. Ella me ha comentado que gran parte de su misión es transmitirles el entusiasmo que ella siente por la ciencia para que los jóvenes a su vez se entusiasmen y puedan seguir el camino de la ciencia. No quiero alargar más la presentación para que ella nos dé su conferencia, y pues les pediría que aplaudiéramos una vez más su esfuerzo por estar aquí.
Good morning. My Spanish is not strong enough to give a talk in Spanish, but I assume that you all understand English and you will understand me. And I want to ask you if you have questions or something is completely unclear or I'm losing you, please raise your hand and ask even in the middle of the lecture. It's better not to lose you than to answer, if I can answer. Don't think that I know everything. So what I want to talk today is a question that I cannot answer, but I have a suggestion. And this is the correlation between the genetic code and the products of it. The products of the genetic code are the proteins, I think that you all know about it, although formally you're a chemist. Yeah, but it's all chemistry anyway. And I will try to relate to this question from the point of view of our studies and a chemical interpretation of them. May I have a little bit more dark now? Less light? Can I control the light? Okay, so far it's okay. Later we need some more. So you know that proteins perform almost all cellular functions in every living cell. What I have here depicted is just the mammalian human functions, but bacteria also have proteins, will not live without proteins. Flowers, trees, cockroaches, animals, whatever. I think that you know that in life, in nature, structure means function. And I don't think that I have to explain much. I'm using this very complicated machine as my way to explain. This is paperclip. It can keep together a few pages of paper, and exactly the same material, same size, same composition, same everything. If it doesn't have the right structure or right fold, it will not do the job. You know all these very nervous people that play with these things. They don't put them in acid, they don't put them in heat, they don't cut them, they just play when they are nervous and they get out things like this. It's exactly the same thing, but doesn't have the structure, and that's very important also for proteins. So there are 20 types of amino acids, and the sequence of these amino acids, which are the building blocks of the proteins within the proteins, enables the protein function. I don't think that I have to explain this. I think this is by now a textbook knowledge. And this is dictated by the structure of the gene that codes for the protein. So the sequence of the amino acids is determined by its gene, and this is what gives the functionality of the protein. There are 20 amino acids, 20 types of amino acids. In each protein are about 150 to 600 amino acids, and their sequence is determined for allowing their function and by the genetic code. The genetic code is the genes that are coding for each protein. Each protein has its own gene, each gene has its own protein. This is one of the main components of DNA. And a DNA is a string that has these genes, but it has only 4 letters. All our life is 4 letters. When I heard it when I was a school kid, when at that time DNA was discovered, I didn't believe it. I was fascinated, and until now I am fascinated. All life is 4 letters. Just unbelievable. And it is expressed in 20 letters, but it's 4. And if you let me tell you a story, when I ask young children what is DNA, now even 6 years old children talk a language that has DNA. So I ask them, what is DNA? Yeah, of course we know. This is the tool that the police is using to identify criminals. So this is for children. So there are some genes that are specific and give the criminals their properties, but most of the genes are coding for proteins that exist in all of us, that are allowing our normal life. So this is DNA, I think you all know. It's made of a double helical structure that has a backbone that I won't describe today, but the backbone more or less requires double helical arrangement. The bases or the nucleotides are here. These are the language. These are the 4 letters of the language. And they're making pairs. Oh, but give it up this light. See? So if it's C, also this light. See. The fold of DNA prevents reading. And I think that you also know that the bases are making base pairs. That they are, in my drawing, orange and blue and yellow and purple that I use for the letters. And they are packed in base pairs, and the rules that decide how the base pairs are correct for all DNA, but also are correct for DNA interactions with another molecule that I will introduce now, RNA. So RNA is actually like DNA. It has also 4 letters. 3 of them are exactly the same. One of them is a little different, but chemically not important. What is special about RNA compared to DNA is that it can live as a single chain. This is because a little bit different backbone. This is deoxy, DNA deoxyribonucleic acid, and this is just ribonucleic acid. So here the information is available. And as I said a minute ago, the rules of the base pairs in the DNA are like between DNA and RNA. So when a gene has to be translated to a protein, part of DNA, let's say this, is being transcribed to the RNA, which is making the same base pairs as within DNA, RNA-DNA base pairs. And now here the information is available and can be translated to growing chains by a molecular machinery called the ribosome. So I was not only fascinated by the 4 letters of DNA, but also how ribosomes work. So ribosomes exist in all cells. They are universal. They are performing their main functions, the decoding of the genetic code and the making of the bonds that takes care of the proteins. They are doing the same way in all cells. Exactly, well, maybe not exactly, but very similar way in all cells. Both things, the decoding and the production of the protein, which is made of amino acids that are connected by peptide bonds. These are the bonds between the amino acids. And this is very similar in all cells, all species, doesn't matter. Bacteria, horse, flower, everything. There is a huge number of ribosomes that function in each living cell. In mammalian cell, it can contain million. A mammalian cell, one mammalian cell like liver, can contain 5 to 6 million ribosomes working all the time. And in these cells, the molecular weight of the ribosomes is 4 millions. Even in bacteria, they can come up to 100,000. Their molecular weight is 2.5 millions. So the ribosomes evolved and grew with evolution. But the functions that they do are exactly the same, or almost exactly the same in all species. The decoding and the production of the peptide bonds, the proteins. So they act continuously. They can form up to 40 peptide bonds in 1 second. And I needed 6 hours for one peptide bond, a single one. They hardly make mistakes. Their mistakes, it's still not a real number because there is controversy, but between 500 to 1 million for a mistake. So it's very, very accurate. We like to describe them as factories. So this is the ribosome factory. The instructions from the messenger RNA are coming in linearly. They are being read here, decoding here. And when the triplet here, each three nucleotides are coding for one amino acid, the specific one. So when three nucleotides here are coding for this amino acid, it will be going in. So if it's a factory, these are tracks that bring the building blocks. These tracks are also RNA molecules, transfer RNA. I will talk about it quite a lot later. So after this amino acid is coming in, being decoded, and being connected to the newly grown protein inside, the track can go out empty. The protein is longer. Messenger RNA is progressing in one triplet, one codon. And all this needs some energy, so I made it like gymnast. That's the whole thing. Very simple. Huh? Just took 20 years minus 2 weeks to understand this. So I want to talk about the tRNA molecule. I'll come back to it a few times. It's always, every amino acid has at least one tRNA, and the tRNAs are almost the same in all species, bacteria and elephants. They always have an L-shaped structure with anti-codon loop here. You can see it here. 3 nucleotides that will read the codon, and a piece here that is also single strand. So the whole tRNA is double helical, but this end of it, the 3' end, which is carrying the amino acid, which is shown here, this is the one, it's always CCA in all tRNAs, in all species. So this is the anticodon loop, and this is the 3' end, always CCA, and then bound to amino acid or to the protein, growing protein, and it's fully conserved. CCA, fully conserved, in every species, everywhere. So actually, only this part is involved in bonding. Most of the tRNA is involved in recognizing its place and in decoding. So the functional sites are here and here. In bonding, only this part, and I will relate to it many times. In finding its place within the ribosome, it needs all this complicated structure and very long one. For those of you that know what are angstroms, it's 70 angstroms from here to here. Much, much longer than anything that can be recognized chemically. So what happens? Here is messenger RNA. It is made of codons, so each triplet is a codon, and I mentioned them here, I showed them here. Each 3 is this rectangular. On the ribosome, there are 3 sites, A, P, and E. P is where the first tRNA binds, and it binds by codon-anticodon interaction, exactly the same type of bonds that are made in RNA. And on the other side, it carries the amino acid, the cognate to this bond. So these 3 sites are on the ribosome, on the small ribosomal subunit. The first one that is being populated is P, then A, and then E is the exit. This happens on the small ribosomal subunit, which is the upper floor of the factory that you saw a minute ago, the top floor, where decoding happens. And the lower part of tRNA binds to the large ribosomal subunit where it makes the bond, the peptide bond. So let's look at how it happens. But how about this slide? I'm sorry. I'm sorry, the movies that I will show you were made 14 years ago by us. Oops, it works. Together with 2 students from the Art Academy of Jerusalem. And they are now on YouTube. You can see it at home. No. Oh, yeah, good. Sorry. Thank you. I thought that he's going to put on the lights. You can see it at home. So it's 13 years in YouTube, and there were thousands, 10,000s, 100,000s of hits, and nobody complained. So I think that this shows that they are more or less correct. What you will see here is bacterial ribosomes, but since the function is the same, they can symbolize everyone. So what you see here is the small ribosomal subunit, and messenger RNA is coming to it, and it will take it. Have a look here. Pay attention to this point. Here. Look. Oops, did you see? It took it. Very small motion here, but very important. And if you think about this motion is run by all this part. And this part is of molecular weight of 250,000. So it looks very small, but it is not only very important, but also very difficult. Anyway, the messenger RNA is now in place, and it's all monitored by a factor, initiation factor, a factor that is non-ribosomal factor that tells the ribosome to start, initiation factor 3. It monitors and it lets the first tRNA to be brought by initiation factor 2. They monitor each other, and once they are happy, they go out, and the large subunit can come and interact with the small subunit by surface, by complementarity of the surface. And I'll show you something else in a minute, but please pay attention. This is tRNA. It is bound here for decoding, and it carries an amino acid. So here comes the large subunit. It will associate with the small one by complementarity of their surface and by making chemical bridges. Here, look. 13 of them. These are the bridges. So now we have the whole ribosome has been made. You can see it from the solvent side. And tRNAs can come in real quick, 40 times a second, with their elongation factors. They are being decoded here, and peptide bond is being made here. And the ribosome helps in, out, in, out, in, out. Now we took away the large subunit so that you can see what happens when peptide bond is being made. And please pay attention really carefully now. tRNA comes in, being decoded here, and moves sideways. It comes in this way. This amino acid will now be connected to the growing protein, but it goes out. Although most of it moves like this, the lower part changed the orientation. Look, oops, it changed the orientation, the lower part. This part that is the fully conserved, the CCA at the end. So the whole process continues until the protein is large enough to come out and fold, alone or with chaperones. And when there is a codon that says stop, everything is done, the stop codon, tRNA is being replaced by other factors, shown here in gold, which are release factor or recycling factor. So now the protein is very large. It will be large enough to come out in a minute, now. So now the two subunits dissociate. The protein can go and do what it needs. tRNA and messenger RNA and the ribosome are ready for the next job. And that's all. That's the whole story. So if you want to see the two subunits, two ribosomal subunits in higher resolution and in more detail, what you saw in the movie was very fuzzy. So the small that does the decoding and the large that does the peptide bond formation. are shown here. They are made of many components, mostly ribosomal RNA, which is shown here in gray, and many ribosomal proteins, up to 21 in the small one and 34 in the large one. And again, what you see here are bacterial ribosomes. So when we looked carefully on their structure, we found that all the functions, or all the important functions, decoding and peptide bond formation, are produced in environment which is only RNA. I don't have to explain RNA, do I? No. Okay. Only RNA, no proteins. Look, it's all gray here in the decoding, and here in peptide bond formation. This is tRNA that decodes there. All RNA. It means that the ribosome is a ribozyme, is an RNA machine. So what does it mean? Children tell me that this shows that the cells don't trust protein to make proteins, so there are two different categories, and I think it's correct. I think that this is an elaborate way to control that there are two different kingdoms to make more or less each other. It also is in good agreement with the composition of ribosomes. There are 2/3 RNA. Even in mitochondria, where half of the RNA is replaced by protein, still the active sites are only RNA, and they are fully conserved. It also is in agreement with the idea that there was an RNA-dominated world, maybe other things too, but mainly RNA, before life in the prebiotic time, and that RNA that is known to be rather inefficient can become as efficient as in the ribosome. It means that nature has tools to change inefficiency to efficiency. So if we look at what we saw until now, in the functional ribosome, this is the small subunit, the large subunit. Here there is a tRNA. One of them you can see, there are 3. And they are bound to the ribosome in the 2 sites, the decoding center in red, and the PTC. PTC, it's peptidyl transferase center, where peptide bonds are being made. This is what we saw until now. When we look at the composition of the ribosome, and let's look at RNA first, we will actually look only at RNA. In the RNA of the large subunit, we don't see any symmetrical region. We don't see any symmetry. When we look at the whole ribosome, we don't see any symmetry. Yet, if we look at it very carefully, we find that structurally there is a region that has symmetry. Only structurally and only the backbone. This is the region. And actually it's made of 2 parts. It's twofold symmetry here. So if I enlarge it, this is the 2 parts of it, the 2 parts that you saw a minute ago. This will be the green, and this will be the blue, and I will keep it the whole lecture. The green side hosts the tRNA that is in the P site. We call it the P region. And the A site hosts the tRNA that is in the A region. And it's altogether 180 nucleotides. The ribosome has 4,500. It's a very small number. I'll come back to the size in a minute. And essentially, it is correct only for the fold, not for any symmetrical sequence. So essentially, this is the region. You can see here, blue and green. Maybe not good enough, so we filled it up for you. This is the region. And this is a semi-symmetrical region, and it hosts the tRNA. I talked about it enough. So in the ribosome, this region, the blue and green that I showed a minute ago in the large subunit, it is connecting the two hands. You remember in the movie? The two hands that have motion? And it is connected to the other functional site, the decoding, by the tRNA. It's really far away, but the decoding happens here. What you see here is the shadow only of the large subunit. So essentially, it sits in the middle of all the functional components. The two hands in, out, and the DNA, the RNA. Now think about it. RNA has to move, messenger RNA has to move by 3 nucleotides when the peptide bond is being made. How does it know it? It's so far, there is no chemical way to transfer, so simple to transfer this notion. Also the hands, one hand has to bring in and one hand has to take out. And the distance between here and here is 200 angstroms. It's impossible just by normal van der Waals or chemical bonds. So we think that this is the middle of the symmetrical region is the transmission center that tells there is a peptide bond being made here, it goes out and bring another one in, and messenger move together. It can send mission to all functional sites. So this, just to remind you, that these are the moving parts in the function. So if we want to look at the size of it, you see here the whole large subunit. The size of this very important region is this. That's it. That's all. We talk about this. And if we look at it in molecular way, the ribosome here is in gray, here and here. The two tRNAs in space fill look like this. Peptide bond will be made here. Or if we show them atom by atom, they look like that. Peptide bond will be made here. That's what happened. And this is why tRNAs have to rotate inside. The machinery, if we want to look at it from the point of view of symmetry, this part is shown here, and I will show it to you larger. So it has the A site, the blue, and the P site, the green. And what you see here is how they accommodate the CCA amino acid or CCA growing protein, exactly made for it. And when I talk to chemists, they tell me that this can be a machine on its own. It has the right size, the right parameters to keep it together and so on. So we suggest that this was, or similar to it, was a machinery in the prebiotic time. So I just want to remind you, this is the tRNA, and we are talking about this part in the machine, this part. The blue from the A site and the green from the P site. That's it. So this is what we are talking about. And from top, if we look at this piece of the ribosome, the 180 nucleotides from top, in all known structures, or all known structures that I knew at that time, but now there are more. So they are here, eubacteria, eukaryote, archaea, and mitochondria. All of them, I put one on top of the other. If we look only at the fold, they're exactly the same. We cannot distinguish between them. And the two-fold is shown here, the two-fold axis. This is the part. And the high conservation of the symmetrical region indicates that its existence is beyond environmental conditions, because it's the same in bacteria and in elephants, in everywhere, exactly the same. Not sequence, but structure. So this gave us the feeling that we are talking about something before the ribosome, from where the ribosome evolved, and we call it proto-ribosome. We proposed that there was a proto-ribosome before ribosomes. And this will be, in our idea, it's a prebiotic bonding entity that is still functioning in all contemporary ribosomes, even in us. In us, there are millions and millions of ribosomes that contain Ps that came from the prebiotic time. or if I write it in a more ancient way, there is an RNA apparatus with catalytic capabilities that functions within the contemporary ribosome. You remember this? This is the machine. So how was it made? Let's look at it again. It's made of two parts. So one of our ideas is that it was made by dimerization of very similar parts. Not exactly similar, but very. So in the RNA well, there were pieces like this, and they knew to fold, and they knew to associate, and they made this machinery that we can see it from the side, and it could make bonds. Maybe many bonds, but also peptide bonds. It means it could make RNA bonds in the beginning. And we have several papers on it. I must tell you that not everybody agreed with me so easily, but after more than 5 years, 6 years, came a paper from Montreal that says the same from another point of view, so now it is more acceptable. So if we look at it from, again, there were two pieces like this. They came together. They had an active site. From the side, you can see it like this. And in this active site, amino acids, or whatever was at that time, could be connected. Not real amino acids, but their precursors. It's built, this machinery is built of pieces that are made of helical stem, elbow, and helical stem. That is the main structural component of the symmetrical region, but also of tRNA. Actually, what you see here is tRNA on the side. So we are trying to make the proto-ribosome. One way is to produce pieces of RNA that should fold according to stem, elbow, stem, like this. So we can compute and produce them, and we have many that we produced. Another way is to take the part from the ribosome and make it. So this is the part. It has the P site and the A site. We just make it, and we try to see whether they will make a little proto-ribosome together. What did we find? First of all, we found that we can get dimers. You can see here dimers. I really don't want to go into details, but we can see here dimers of several of our constructs. What are the several? So here we were really surprised. Until now, I don't understand it. There is a non-uniform tendency to dimerize. Some of our constructs dimerize and some not. The P site dimerize, the A site not, and the P and the A not. So this shows that survival and fitness of natural selection seem to play a major role even before Darwin. It means what we found is pre-Darwinian Darwinism, because Darwin talked about organisms, not about molecules. This is Darwinism in molecules. And because only the backbone is similar, there is no sequence symmetry, we think that maybe in the beginning the ribosome was made from two identical pieces that dimerized and then changed. So this is what I try to say now. This is the way the ribosome looks now. If we put the A site over the P site, it's very similar, but not exactly the same. And maybe these different functionalities came with time. In the beginning, it could have been the same. From proto-ribosome to modern ribosome, going from what we don't know what it was, but presumably a lazy RNA machine, we have now the whole ribosome, and this is what nature did to us. And by adding proteins, and I'll come back to it. But this whole thing is based on the suggested existence of RNA-dominated world. First of all, we have to suggest that or to believe in it. And on the finding that RNA can replicate and elongate itself, and it can become later on an entity that can make life evolve around it. So now there is a logical problem. Why should RNA make enzymes from proteins that are better than enzymes from RNA? It means made competitor better than you. We don't think that RNA decided to make competitors better than it, but it happened. If there was already a machinery, the amino acids snatched it because they're so clever. When they showed up, they snatched it to make proteins. But it seems that there was a pre-ribosome. And ongoing binding experiments show that it could be. We have several constructs that can bind RNA precursors or molecules. So now we can come to the question how the genetic code emerged. If we take into account all what I told you until now, we can ask this question. And the other way to ask it, what was first, the genetic code or its product? Why should be a genetic code if there are no proteins in the RNA world? How can proteins be if there is no genetic code? So it's still not clear, but what was first? So we think that the initial dipeptides that were made by this machinery could be the substrates for the formation of next protein bond. So this way, oligopeptides could be made. And among the oligopeptides, those that were either useful or could make good things for themselves, well-performing, they survived. What do I mean being useful or make a better machinery, so on? So this is the same thing, the existence of well-performing. Well-performing small oligopeptides is shown here. So the oligopeptide is this, and it has 3 histidines. They could carry a metal, and the RNA world needed metals to be brought. So this is useful, and these are those that could stabilize the machine that made them in these positions. And we suggest these positions based on the structure of the modern ribosome. So this suggested the genetic code was created by or according to its products, which were found fit and useful and therefore survived. This led to the creation of primitive original genetic code, which co-evolved together with the products and with the ribosomes. So if I repeat it, the genetic code co-evolved together with the ribosomes as well as with the products. So it let us ask I will jump this. It let us ask a question. Oh God. What was first? The chicken or the egg? The genetic code or the proteins? So what was first? Anybody here can answer? Genetic code or proteins or chicken or an egg? I will have a question to any of you. Because we think that really first was the ribosome. The proto-ribosome, the small ribosome, but at that time there was neither genetic code nor proteins. It happened on this machinery, the proto-ribosome. So I could stop here, but because there are some excitements recently in our lab, I want to tell you about them. And this has to do with antibiotics. I will talk about it much more tomorrow, but a few words now, a few minutes. Because of the fundamental role played by ribosomes, many antibiotics target them. Half of the useful antibiotics are targeting ribosomes. It's very clever. The next generation of the bacteria that we want to get rid of will be if with less proteins or with wrong proteins, so at the end the bacteria will die. Natural antibiotics are the weapons that bacteria from one type use against bacteria from another one. It means the wars in the microorganism era, in their little world. Question is, how do the tiny antibiotics paralyze giant ribosomes? Ribosomes from bacteria that we want to get rid of are about 2.5 million Daltons, and antibiotics less than 1,000. And the trick is to target functional sites. So what you see here is the skeleton of the large and the small subunit with these circles of them, and each circle is representing a family of antibiotics. It means natural antibiotics that were improved by companies, and each of them, or most of them, are sitting in functional sites, either in the decoding, or in the bonding, or in the tunnel, or in the hinging these motions. So if even one of these functions is stopped, the whole process is stopped. This is what antibiotics are, natural antibiotics, bacteria against bacteria, or another microorganism against bacteria. So instead of talking, I have a little movie, as before, made in the same way, the first one, and you can see here adeine, that is disturbing messenger RNA binding. Look, you don't have to be large. Just sit in the right position and hamper the binding. Second one that you can see is tetracycline, that occupies the position of the A site tRNA. Just occupies the position, so when the A site tRNA comes, it cannot bind. So the third one that you will see is working on the large subunit, and it's one of the first being used medically. So it's called erythromycin, and it sits in the tunnel where peptide bond is being made and protein should go, and right in the beginning, 5 to 7 bonds, and it's already disturbing the motion along the tunnel. And the fourth one is clindamycin, the chemical cleverest one. It goes into the position where the bond should be made between the amino acids. Choop, see, in the bond. So there will not be a protein. So these are four examples, and we learn them by using crystals of complexes with antibiotics from the ribosomes were from bacteria that are pathogen models, not real pathogens. Because we cannot grow real pathogens, we had lots of problems. So we learned about a lot. We learned about common properties of binding antibiotics, but not on the specificity of the pathogens. So therefore, we crystallized ribosomes from a pathogen. What you see here are crystals of the large ribosomal subunit from Staph aureus. So may I tell you a story about this line here? You see it here also? Anybody knows what it is? No, you cannot, because this is the eyebrow of Zohar, our student. And only Zohar has these eyebrows. So everybody in the lab, when they try to crystallize and could not, they put their eyebrow, nothing. Zohar, come and give the eyebrow, she gives, crystals grow. So there is something in Zohar's eyebrows, but I just want to tell you one of the tricks of getting crystals. So what did we study from it? We studied lots of lessons. I will show you only a few of them. First of all, pay attention. This is the bacteria that we use, is Staph aureus. It's a bacteria that can make multi-antibiotic resistance against many antibiotics. A very, very nasty bacteria. So first of all, there is now a common belief that better antibiotics cannot be done. We learned from it that it can be done. A company in Vienna made a new antibiotic, I can't talk about the details now, that is 16 times stronger than the normal ones. And this is just by adding one hydrogen bond, this one. So from the other side, this. Just one hydrogen bond makes 16-fold higher potency. The name of the company is Labriva Therapeutics. Lesson number 2, 3, we want to talk about number 4, which is much beyond my expectations. I didn't even dream about finding this, and I want to show it to you. I'm sure you cannot read it, but you can understand that what I'm trying to show. These are many bacteria, and protein L free, large subunits free in all of them, all of them. They're all more or less the same. They're very, very similar, except for here and here. Here there are differences. There are insertions in 4 bacteria. And if we look at them here, these are the 4 insertions. And all 4 of them are pathogenic. The rest are not. They are pathogenic. It means they cause diseases. So this is a specific region that is found only in pathogenic. That's really very exciting because we can think about this as targeting just the pathogens. And even more convincing is what I'll show you now about the RNA. So this is the RNA the cyan is the RNA of the pathogen, Staph aureus, and the model is the gray. Now there are 4,500 nucleotides in every ribosome of bacteria, more or less 4,500. Here there are 9 additional ones. When one looks only at the sequence, nobody sees anything. Who cares for 9 out of 4,500? It's much smaller than expected standard deviation. Even 20 of them did not make any importance or did not make people think. But when you look at the structure and put one on top of the other, you can see that wherever the sequence is exactly the same, there is no difference. But here there is this additional part. And this additional part can be a new target for antibiotics, like here. So something that the bacteria didn't think about. It's not directly in their functional site, but it is important for function. Let me show you. We identified, first of all, I want to show you that we identified many. That's first of all, because I really love to look at it. All these bluish pieces are new, are Staph aureus, and we looked about this one. The second, I want to show you how it looks when it stands. So here are the new ones from 2 different views of the large subunit. And the third thing that I want to show you, is that, which I don't have the slide here, is we identified 25 like this, and 16 of them are, when we block them, and we block them with antibiotics that can be chemically monitored and chemically improved and chemically optimized, 16 of them stop the ribosome function because they're either important for the interaction with the small subunit or with other ribosomal cellular complexes, components. So they are not directly in the active site, they are not directly in the peptide bond formation or in the decoding, but they are important for life. And the bacteria, it will take the bacteria time to learn to make resistance to them, but much, much longer than for a real active sites, because there is no sequence of genes in any bacteria that knows to make a resistance or to target these places. So this is something that we are now suggesting to make, and I'm really excited about it, because until now I thought there is no future to antibiotics. There is resistance and that's it, and nobody can think about new ones, but these are new ones. Also, there is an environmental consideration. This is our lesson 5. So most of the antibiotics that are used now have a core and extensions. The cores, here there are some cores, not all, but some of the ribosomal antibiotics cores that are not degradable. They are not digestible, they are not consumed by the body, and they are not degradable when they come out to the sewage. So they are contaminating the environment. And when they are in the sewage, they can go to the water system, and grass can be grown on them, and cows are eating the grass, and we are getting them through the milk or the meat of the cows. Or I just brought one example. But these are contaminators that even when there is an overuse of the antibiotics, even when antibiotics are used in a very restricted way, there are still components that are coming to us or to the animals and will cause more resistance. So the new antibiotics, those that I suggested around these new positions, can be now made of degradable compounds, like oligopeptides, like complementary DNA, like PNA. And this is what we are trying to do now, and they can be optimized by us, first even without the cells, just in vitro. And in vitro, we showed, as I told you, 16 new potential sites. So in summary, the new insights obtained from these high-resolution structures of the ribosome from a genuine pathogen provide unique chemical tools for suggesting novel sites, I showed you, for suggesting how to improve clinical, I didn't show, for better distinction between pathogens and useful bacteria. This is by making, now we can make pathogen-specific antibiotics, because now we know what is in the specific ribosome that I talked about, but we can do it for every ribosome. And actually this week came another paper for the same Staphylococcus of the whole ribosome with very careful, it came from Strasbourg, with very careful alignment of the pathogen on a model, and there are more sites of this type that may be used. So, well, sorry, I don't know why this is here. Now I can stop, I can finish, but also I can tell you how we started. And the chairman has to tell me what to do. Are not you tired? No. So, what was known when we started? It was known that proteins are the cell workers, they perform almost all tasks, that they are doing it because they have a structure that is determined by the genetic code. This was known, but not how. It was known that ribosomes make proteins. I didn't discover this. but not how they function. And when I say that I want to understand this, they say you must be kidding. It means they made jokes about me. They said it nicely, like I'm laughing, but they really made lots of jokes about me. I was the dreamer, the fantasionaire, the fool, whatever you want. They say that the interior of the ribosome is a black box, and that's it. I wanted to understand, and for this we need crystallography. For those of you that don't know, there are crystals, must be crystals for crystallography. Oops. I can tell you about new ideas later, but this is the basic. The X-rays, Röntgen rays that are going through these crystals are diffracted, and we get diffraction pattern. We get it in all the directions around each crystal, 360 degrees around it, the whole sphere. So we have to collect them all. You can collect it either on film, like in here, or on detectors. And these spots are where each of the X-ray beam came out. And the further they are from the center, the higher the detail, what is called resolution. So they are all the diffraction from all directions are merged together by computers. It's not so automatic, so we still need, you see this little thing? This is us, the crystallographers, like me. So these little things have to still to think about all types of things that I won't go in, like in here. And at the end, you can see an electron density map. The X-rays interact with electrons. It's like a topographic map that shows mountains, and we think that in every center of a mountain there is a nucleus. It means this is an atom. We can combine them. So this is a carbon type molecule. Here there is a metal. You can see that there are more electrons. So we can do it for small molecules. Now we can do it for bigger ones, even ribosomes or proteins. This is crystallography. For this, we need crystals. Now, everybody of you is using salt in the kitchen or in salad. It's made of 2 atoms, and it has a natural tendency to produce crystals. When we buy them, these shiny little things in the salt are salt crystals. Crystals can be described as entities that have full repetition, full periodicity in 3 dimensions. This, and this. And you can see these are the chlorines, and the sodiums are inside. So these are crystals of salt. We know about crystals of proteins, but this that we know now, they are ribosomes, crystallized this. There was a huge, now we know, at that time they didn't even know that it's like this. There was a huge question mark. And the question mark was because many very, very distinguished scientists with very good labs tried to crystallize ribosomes and failed. Even Watson, the Nobel Prize winner, tried for 10 years and failed. And they explained the failure by high degree of internal mobility, considerable flexibility, functional heterogeneity. It's all correct. We now know it from the crystal structure. Chemical complexity and large size. This is all correct and asymmetric. They overlooked, in my opinion, the importance. They knew about it, but they overlooked the importance of the tendency to deteriorate. What do I mean? When one produces ribosomes from bacteria or from any cell, very carefully, the best careful ones, we can get only 50% of the expected activity, even less than 50%. And this is called good preparation. So I thought, when I tried to make it, and I heard that everybody, and I even went to Berlin to study how to do it best. So when they had 51%, they were really happy. I thought, what happens there? must be that at least half of the preparation is either deteriorated or has a own structure or something that doesn't let it work, which means that there is no chance that there will be crystals if you start from a preparation that is not homogeneous. This is what I thought. I didn't have an idea what to do, but this is what I thought. Then I was lucky. And my luck was a bicycle accident, that a very, very severe accident that I got when I was riding my bicycle without a helmet. You know, this helmet that protect the head. At that time, there were no helmets. I'm talking about 1978, so 79, sorry. So I fell off and I had a very severe brain concussion that prevented me from coming to an experiment I don't want to go into all the details, but when I recovered, I had time to read, because I was not strong enough to read science, but I could read stories. So I read about a delegation that went to the North Pole, and so how they wanted to find out how the bears, when they sleep, the winter sleep, how do their metabolism work. And this is what they wanted to see. And side finding for them was that inside the inner part of the cells of each bear, the inner part, on this, ribosomes are making monolayers, periodical monolayers, something that scientists try to make for over a century. And every bear, every cell, every winter has it. So this, first of all, showed to me that ribosomes can be orderly packed. And second, I thought, why? Why does it happen? Because they didn't think it's important, and they had a picture, an EM, that was really very low quality. I got excited about it, because I thought, why does it happen? And in my opinion, bears will get up at the end of the winter. And then they want to do all types of things that they didn't do when they were asleep. And for this, they need proteins. And for proteins, they need ribosomes. And this is the way, the mechanism that nature provides to maintain non-deteriorated ribosomes that are active. Otherwise, they won't be able to function. So I thought, I learned from it, or I assumed from it, that stress, oh, you can see another bear. He also has his ribosomes organized. So I thought that stress induces periodic packing of ribosomal particles, that this is the machinery that nature provides to maintain activity. And if so, why not to try to use ribosomes that are from bacteria that grow under stress? So you don't have to read all, but these are what you used. Dead Sea bacteria, hot springs, and waste of nucleic facilities. So the Dead Sea is in Israel. It's the lowest part in the world. You can see it here. I was working on it when I was younger a little bit, not much, but I knew quite a lot. So this is the Dead Sea from top. This is the way you look at it. You see it from close by. These are salt structures that came out when the sea evaporates. So the sea has 3 molar KCl and over 1 molar sodium chloride and barium and magnesium. And the temperature in the summer can reach 50 to 60 degrees Celsius. So there is a lot of evaporation, and you see here the results of it. There is no life. It's called the Dead Sea, and do you see life here? But there are 2 bacteria and 1 algae that live in this sea. You see here? These are colonies of Heloarcula marismurti. And you see it on the salt that came out in the evaporation. You can maybe see it even better here. See here, colony, this pink here too. The colonies stay even when there is evaporation. So what you see actually here is the water level in January was here, December was here, and in September when this picture was taken, 70 centimeters of evaporation, it's just enormous. And bacteria still exist. So there are actually two bacteria, Haloarcula maris morti and Halobacteria volcani. So we took the ribosomes from this side, or this one. Also we took ribosomes from this really crazy but wonderful bacteria, the Anococcus radiodurans. The Anococcus radiodurans is a bacteria that can live under super extreme conditions, very hot, very cold, environmental dust, hunger, and irradiation. It was found in sewage of nuclear facilities. And this is actually very, very similar to the pathogens and bind the antibiotics similar to pathogens, whereas this is an archaea. So it's not exactly the best model for antibiotics, but it serves also at this. So in the internet, it says, they quote one of my papers that we made 25 different conditions, not 25,000 different experiments. You can see here from the book, I took it, experiment 29. So we could make 1,000 different conditions in one experiment, very, very simple. But anyway, we got microcrystals, which grew after several years to wonderful crystals. You can see here 6 types, we have 25 types, and all were of ribosomes, ribosomal particles, subunits. So after 6 years, I should tell you, we started in the last week of 1979, in December last week, which let many people write in their report that Ada started her crystallization in the 70s. But it's correct for the last part of December, the 79. But actually we started in 1980, and in 1986 we had good enough crystals to go measure. I think that you know that measurements of crystals are done by generators, and the best generators for complicated things are synchrotrons, accelerators. Here is the synchrotron in Grenoble. And it's an accelerator of particles that works according to particles are being, that's me? Are being accelerated here, run around, and in the tangentials we can put stations, like measuring station, like here. So we went to a synchrotron, and what we got, wonderful. Here you see this Haloarcula, large subunit, in 1986, wonderful diffraction to the edge. Now you had to see the students. There were already a few students, and they jumped, yeah, yes. So they were only 2 or 3 years. I was already 6 years in this game, but I was also happy. You know for how long? 1 shot. The second was gone. In 1 shot, we lost everything we had, and as I told you, we needed the whole 360 degrees rotation. So in this 1 shot, we didn't have even 1% of what we needed, even less than 1%. So this meant that we will need hundreds and hundreds of crystals for a data. And because each crystal may be a little different, maybe we will need thousands of crystals. And this is not a practical way to go. We repeated it over 100 times in 7 different synchrotrons and got all the time the same. Crystals decayed after one shot, one experiment. So this was good time to feel like. We started here, you see, here we were. And we saw this big mountain, only this big mountain. We didn't see this one. We started here. we thought that this is the Everest. So we started climbing it, and climbing, and climbing, and climbing, for 6 years, until we reached the summit, just to discover that there is a bigger Everest behind. And we will have to get there. Crystals are not enough. So this was a good time to stop this project. And if I stopped, I probably couldn't talk to you about it. But it was also possible to think, why does it happen so badly? So all crystals of biological systems decay in the beam. And all biological tissues decay in X-ray or in Röntgen beam. But this was the worst situation. So why? And I cannot go into all details. I just want that you pay attention to these 2 lines in the middle. I thought, and I think it's right, that the decay is a consequence of primary and secondary events. The primary is the local disruption of bonds by the X-rays. X-rays can't cut the bonds. CC bonds, CN bonds, they cut. But then, after the bonds are cut, there are 2 atoms. Each of them is unhappy. One is unhappy because he has an extra electron, and one is unhappy because he lost its electron to the other one. So they are both unhappy and they run around. This is what we call propagation, looking to satisfy themselves. And meanwhile, they break more and more bonds. So we thought that this is the process, and we thought that we cannot fight against the primary reason, the disruption of the bond by X-rays. But we can minimize the propagation by not giving enough energy. And this is what we did. We flash cooled the crystals to cryo temperatures, -200. I wrote the exact temperature. If somebody wants, I can explain why, but it's not important. So this way, there is no energy for propagation, and the deterioration became much slower, almost none. So here is the experiment. We did it in Stanford. And important for me is that you look at my face. That show that not only everybody told me it won't work, I was also not sure. And that you see Hakon Hope, who was our collaborator, and with him we did the first experiment. So everybody told me it won't work. Even my good friend came and said, do you think that something will come out from this, whatever? But it came out. Wonderful, huh? Isn't it? From these crystals and from others. And look at my face 20 years later in Grenoble with the machinery that we used to cool the crystals and the weld. So what you see here is the number of structures that were determined versus the time, the year. Now, we know the number because everybody that wants to publish a new structure that was done crystallographically has to deposit the coordinates in the Protein Data Bank, PDB. So we know how many depositions were in each year. This is clear. Maybe there are more that were done by companies. Companies don't publish, so they don't have to deposit. But what was published, and you see here the statistics to 2011. In 2011, there were more than 50,000 structures. 99% of them were done at cryo temperature in our procedure. It became routine around the whole world within 4 to 6 months. Even not a year, half a year, and everybody used it. Now, in 2016, there are 87,000 structures, and again, 99% of them, almost all, were done at cryo temperature. So this makes me really happy. It also made the committee in Stockholm happy. So it's time now to thank my supporters and those that let me work, the president of the Weizmann Institute, Michael Sella, who is an immunologist, decided that I can stay, although I was a dreamer and fantasioner. They were supported by the Scientific Advisory Committee, Andrew Anfinsen, Nobel Prize winners. Andrew determined the first structure of a protein, hemoglobin, myoglobin. And Alex Rich, who doesn't have the Nobel, but he was one of those that tried to crystallize ribosomes and failed, but still was very supportive, so I'm thanking him twice as much. They say that Michael Sella, the president, was biochemist, therefore he didn't stop me, but he was replaced at the end of his term by a physicist, Chaim Harari, and he also let me go on. So I'm really very thankful. It all started with a very strong collaboration in Berlin with Professor Wittmann, the head of the Max Planck Institute for Molecular Genetics, that he was really interested in ribosomes. So 240 people worked in his department. Having also me, it was not a big thing for me, so therefore I could go. But when we started to get the good results and I could show that we have diffraction, he was very important in starting a research unit for us in the synchrotron in Hamburg. So unfortunately, he died 10 years before there were structures, but we could work also in Hamburg. So I had 2 labs working at the same time. And I want to thank the research groups in the 2 labs, one in Hamburg and one in Israel, for their determination in good and in bad times, more bad times than good times, but they were still determined. The group in Hamburg was terminated in 2004. And I just want to show you that the technical assistants thought that only angels can work on ribosomes. And the whole group came to the Dead Sea to look for better bacteria. They had better time. And the group in Israel, which is still functioning, is what you see here is a little bit more than the group, also some of those that already left. Is being run by Dr. Anat Bashan, not by me. I'm entertaining you. She's working. And I cannot show you everything about everybody. This is Zohar with the eyebrow. And I just want to tell you, at least the female young student here, that still hesitate whether to go to science or not. Anat Bashan, Dr. Anat Bashan is a fantastic scientist. She has 3 children and very, very happy family. Ella Zimmerman, who is also a fantastic scientist, has only 2 children, but very happy family. Raz, that traced the RNA in the 2 ribosomal subunits, has also 3 children, but he doesn't belong. He's a man, so we talk only about girls. Ilana has 4 children, and she found the symmetrical region, the topic of this lecture. But please look at Tamar. Tamar came to us 16 years ago for 10 weeks. This is very long, 10 weeks. And she had a birthday when I took this picture. She baked a cake. Can you see the cake? Maybe you cannot, so I will show you the cake. Here is the cake. And this shows that my group considered ribosome as sweet. I also want to thank my family, many of my family, my mother, my sister, and so on, but you can see here only my daughter, who is an MD, a doctor, and my granddaughter, who was a school kid at that time. She is the one that crystallized the protein. And she, when I got a prize in Paris, she made a speech. My name is Noah, Ada's granddaughter. As you know, she is a very busy scientist, but she always finds time for me. Girls, it's possible to have a good family and to be scientists. If you hesitate, this is possible. I won't say anything else, but if you love science and you love your family, it works. She gave me a prize, my granddaughter. You want to see the prize? This is the Nobel medal. Her prize is for me as important. The grandma of the year is Ada Yonat. So when I asked her which year, you didn't write a year, every prize has a year. You know what she said? You have to reprove yourself every year. That's the only prize that can be taken from me. And she said, if you don't prove yourself, I'll take it off the wall. And it's already 10 years, and it's still on the wall. So I want to show you that ribosomes became popular in Israel. This is a carnival. He's me. You see? Yes. And he's dancing with his wife, who is a ribosome. Protein comes out of her. And I want to show you what happened to me. So first of all, 2 years ago was the year of crystallography, and there was a paper about crystallography. And one of the figures was about protein crystallography. And they show here the two schools of protein crystallography, the British in green, in blue, and the American in red. And look how many Nobel Prizes came out of it. All these are Nobel Prizes. I think it's the most populated. So this is Bernal, Sir Bernal, and this is Linus Pauling. They themselves have a Nobel. And if you look for me, where am I here? Down there, yeah, here. Treeless. Until now, I'm a treeless person. There are only 4 of us. And this is what an artist made out of me. And if you look carefully, small subunit, large subunit, twofold symmetry, not exact, but very, very, very similar, and positions for new antibiotics in his mind. So thank you.
Yo no sé nada de toda esta sabiduría, pero la alegría de estar aquí y compartir con una extraordinaria persona esto es para agradecerle por siempre. Gracias, maestra.
I almost understood it.
Okay.
Almost, but maybe you tell me better.
She's very grateful with you because she is working in another field, but she thinks that you are very amazing, and it was a wonderful.
Thank you. Gracias.
Thank you, Ada, for your enlightening talk. I was amazed about this symmetry that you found in the large subunit of the ribosome. And I was wondering, this proto-ribosome is only on the large subunit. Have you looked anything similar at the small subunit, at the decoding center of the ribosome? Is there something similar there?
Yeah, the similarity is that they are made only of RNA, both of them. But symmetrical and so on, there is not. And also the small, it cannot be because it is a running process.
It's different, yes. But then you think that it evolved much later than the large subunit, the small subunit, or they
Yes, I think so. I think so, but not everybody. You see, it's only thinking. What we could show is that there is this region that can make bones.
Yeah. Thank you.
Okay, so thank you. It was pretty impressive. My question is, as a woman and as a student who's struggling to be here in this chemistry school, what were your problems or your struggles being a woman and being from Israel and getting the first Nobel Prize as a woman from Israel? What were your struggles? And also, how different it was your life after getting your Nobel? And congratulations.
So you asked many questions.
Sorry.
So I will first talk about the Nobel, afterwards about gender. It was wonderful to get the Nobel Prize. It was a real nice dream. And many things changed in my life. Most important is that I can talk to many young people. They come to listen and sometimes even ask questions, as you can see. I could give lectures earlier, but it's not the same echo. And I also can talk to scientists and to policymakers to try to change. I think we made a big change in antibiotic field. But all this is nothing compared to the feeling I had when I got the discovery, when we understood the structure. This was years earlier, 9 years earlier. Then I couldn't sleep, I couldn't do anything, just think about the ribosome and how it works. It overwhelmed me. So the prize is fantastic, and I have many prizes, and each prize is great. And it's nice to talk to people that otherwise I wouldn't meet, and to see that even young people think about science more. But the discovery was something zillion steps behind. Okay, so how to be a woman, you know. I don't have to say. How to be a woman in science. It requires two things, loving science and loving family. If these two are fulfilled, and there is a good question that really, really interests you, that you are really curious about, then I don't see a big difference between women and men. Well, maybe I see. Men. Can you be pregnant? Can you deliver a child? No. So this is our fringe benefit. Although everybody says it's a lot of work and it's difficult. No. The excitement and the feelings and the involvement, the ties to kids and family are matchless. So it's just a question if you love them both, and then things go on.
Well, first of all, thank you for being here. And I would like to ask you, what would you recommend to those students who want to dedicate their lives in a future in science? I think that sometimes we don't know which are the most important factors in which we should focus on in order to be good scientists in a future. So I don't know which recommendation would you give us to be good scientists in a future? Which are those factors that which are the most important?
There is a difference between the first part of your question and the other. To be good scientists is one thing, and to succeed is another thing. To be good, what does it mean to succeed? We cannot discuss it all. To be a good scientist, I think it's personal. It's how you feel. If, as I told you, I was 20 years the joke of the world, the village fool, but I knew that we progress, and I had good feelings. It was difficult, but I get good feelings. To be successful, how to measure success, I don't know. And actually, to give advice how to find projects and so on, what you asked, I have only one advice. And my advice is, don't ask for advices. Do what you feel is good for you, interesting for you, you are curious about, and you're mad about doing it. This is my advice.
Thank you very much.
Don't ask others. Everybody has its own. And the best is to progress as you are.
Thank you. I appreciate your answer.
Bueno, primero que nada agradecerle a la doctora por su plática, fue muy interesante. Y bueno, la pregunta va dirigida un poco hacia el diseño de antibióticos. Me llamó mucho la atención esta inserción que hay en el ribosoma de Staphylococcus aureus. Y la pregunta va dirigida en el sentido de que pues actualmente sabemos que los microorganismos adquieren resistencias a los antibióticos y cuán factible es que un microorganismo para adquirir una resistencia a un antibiótico realice una mutación en su ribosoma y qué tan permisible es esta mutación, o sea que después, dada la gran conservación del ribosoma, si esta mutación le permitiría subsecuentemente tener un ribosoma funcional y bueno, qué tan posible es que un microorganismo use esta estrategia para hacerse resistente a un antibiótico. Esa sería la pregunta.
You talked about natural mutations, or how easy it is to do it in the lab? Natural resistance. I don't know how many trials each bacteria has until it finds itself to be resistant. Maybe zillion of them, millions of them. Lifetime of bacteria is between 10 minutes to 4 hours. So the slowest is 6 generations a day, and maybe many more. So how many times they make mutation until one of them survives, I don't know. I don't think, they just make. So I don't know how easy it is for them, but for us to copy something like this is really difficult, because we think that we understand. And we cannot have generations of people. It talks about hundreds of years.
We can make a certain number of random genetic codes, and maybe all these random codes existed together a long time ago. And there are studies that show that there are codes these codes are more stable than the actual. And the question is basically why this code in evolutionary terms. I mean, if there are codes that are more stable, why this code is chosen?
So you're asking about the stability of genetic codes? This is your question, if I understood you right?
The stability and, the stability in transcription. I mean, when you talk about, I don't know how to say in English, bamboleo, there are codes that its products are more stable. And why this?
Okay,
why this
I can
genetic
stop you now because now I understand what you are asking and I cannot answer that. That's not my specialty. I can only tell you what's written in Google. But then you can read yourself. Maybe more than Google in some papers, but then you can read yourself. It's not I have no experience in this. I don't study this, I'm sorry. I can't do everything.
Well, thank you for being here. I want to study some genetics things. My question is, you tell us that the ribosome structure in the life is similar, but in one species like elephants, only elephants, is the protein structure the same in all of them, or they are small changes?
You're asking about the ribosomal proteins?
Yes.
Yeah, there are changes. They are, the RNA is much more conserved, and the RNA changes with evolution in big blocks. Proteins, there are differences between them. I showed one protein, and I showed lots of bacteria, and each of them is a little different. And the differences are not only by evolution, but also within the same kingdom. I think proteins are more different, or there are more variability in the proteins than in the RNA. The proteins are essentially not directly involved in any of the ribosomal functions, but they are allowing for the ribosomal functions, for maintaining the RNA structure. So if I go back, maybe I can show you a slide, but meanwhile I can answer other people. No other questions?
Yeah, hi, Ida Jonat. My question is, how do you solve the phase problem in your first structure of the ribosome?
Did I solve the what?
How do you solve the phase problem in the structure?
For the first structure?
For the first structure.
Using multi-metal complexes.
Okay, and well, nothing.
The second was much easier.
Thank you. The catalytic site in the ribosomes of all the organisms is different, but no, it's the same. But the proteins or the other proteins are the same.
Each protein has its own sequence and structure. So you're really interested in proteins. I will show you a little one of it. So from inefficient RNA enzyme, like all the ribozymes, there are about 16 or 17 RNA enzymes today. And then there is an amazingly efficient contemporary ribosome. This happened by the addition of proteins. So you can see here all the proteins in bacterial ribosomes, in the large subunit and in the small subunit, all of them. They all have very small domain, globular domain, and long extensions that go into the structure. Like I'll show you 4 examples of the small subunit. We'll talk about these 4. And look, it goes into the structure, very, very much in. All the 4 of these ones that I show here and here are busy in function. For example, in allowing for function. For example, these 2 here are reaching almost, not exactly, but the decoding site. Here is the decoding site. And they start here. So a little mutation here in this protein does still allow decoding, but the decoding is not perfect, is not following the gene exactly. So it just changes the structure of the RNA in the decoding, not really disturbing it. So this is what the proteins are doing. And they are more and a little bit larger with evolution. Okay?
Small recognition.
Thank you.
Thank you very much.
It's fantastic. You took this and you made it red instead of blue.
Yeah.
Fantastic. Can I get one of the big ones?
Yes, of course.
No,
of
not
course,
this.
the big one. If you want the big one, you can take it.
No,
The small one. Okay, perfect.
whatever you have,
No problem.
big or small.
Ada, do you think that you can send some posters?
Yeah, what I should what?
Sign?
Yeah.
A ver, nada más con orden, por
None
favor.
of this is mine. Yeah. Thank you.
Haciendo la fila. Nada más con orden, por favor, para que...
How do you put it off?
Okay, yeah. Okay, that one. Yeah. But maybe we can fold it very well. Okay.